Hey everybody, so this is the video for the Unit 1 study guide, and so this is going to go over just the general information that you need to know in order to do well on the test. Now, hopefully at this point you have already completed this study guide, okay? This video is not intended to give you the answers for the first time so that you can type them in while you're watching the video. Rather, this video is intended for you just to check to make sure that your answers are correct after you've already answered the questions. So if you have not answered the questions, pause this video, go answer them, and then come back and make sure, and then watch this video just to make sure that you got them right. If you're just typing things that I'm saying, you are not internalizing that information at all. You are just typing words, okay? So let's go ahead and start. So this video is not super long, and I am going to go a little bit fast because where this is a video, you can always pause it, go back and watch it again. So number one, it says describe the process that is responsible for the growth of living organisms as well as repair of damaged tissue. What process is that? That process is cell division, also known as mitosis. And basically what that is it is the copying of a cell into two, now this is very important, genetically identical daughter cells, okay? So let's say, for example, you are repairing tissue. You've got a cut on your hand, and so what that means is you've had some skin cells that have been destroyed. And so we need to repair that damaged tissue and fill that in. And so what will happen is all of your other skin cells in the surrounding area will start dividing to replace the ones that were damaged. So you will go through the process of cell division, prophase, metaphase, anaphase, telophase, and then cytokinesis. And it will do that until that wound or that damaged tissue has completely been replaced. And it's so important that those are genetically identical, because remember, your genes determine the function of your cells, because your genes hold the instructions to make the proteins that are like the workers within the cell. So if you have different genes or different cells that aren't genetically identical, then they're not going to be able to perform the same function. And if you're replacing a certain type of cell, like a skin cell, you want it to function as a skin cell. That makes sense, right? So number two, briefly describe the process of DNA replication, including the enzymes helicase, DNA polymerase, and DNA polymerase. Why is it so important that this process is done correctly? And I kind of just touched on that answer before. So if you remember back from the gizmo, you built your original strand of DNA, right? And then an enzyme came through and split your DNA. So remember, this is happening during the S phase of interphase. Your DNA is being copied, and so when your cells get the signal, okay, it's time to copy the DNA, the enzyme helicase will unzip your genes, okay? So it will unzip it right down the middle, just like a zipper on a jacket, okay? It will split your genes, and what that does is it exposes the nitrogen bases, those letters that are on the inside, so that those letters can then be copied. Now, what enzyme, and remember, an enzyme is just a protein that does a special job. So a helicase is actually a protein. DNA polymerase is a protein. Anytime you see something that ends in ACE, usually that's an enzyme. So once your genes are unzipped, unwound and unzipped and opened up, your DNA polymerase will then read the exposed nitrogen bases, which remember are A, T, C, and G, your adenine, thymine, guanine, and cytosine, and will copy each strand of DNA. Not Dan, DNA. What you will end up with is two identical copies of DNA. And so remember, helicase unzips it, DNA polymerase, anytime you see polymerase, it's building something. So it's building the other sides of the DNA. And so what you end up with is two genetically identical copies. So why is it so important that copying this DNA is done correctly? Well, remember up here, this must be done correctly so that each new cell receives the same genetic information after mitosis. Okay, it's super important that each cell gets the same genetic information, that they become two genetically identical cells. Okay, so describe cell differentiation. So cell differentiation is simply is when protein, let's phrase it this way: specific proteins are made within a cell to give it a particular function or job. So, for example, your red blood cells need the protein hemoglobin, but your skin cells don't need to make the protein hemoglobin because their job is not to carry oxygen through your bloodstream. So, while your skin cells have that information in its genes, all your cells have the same DNA, it's not going to read the genes for hemoglobin, and it's not going to make the protein hemoglobin. So cell differentiation is where we make specific proteins to give cells their special jobs or a particular function. Now, without this process, as complex organisms, we would not be able to have, I can type here, we could not have specialized tissues. Okay, tissues are just bundles of cells all working with the same function. So you couldn't have skin tissue that was different from your muscle tissue if it weren't for this process of cell differentiation. Okay, number 4, what are all the ways in which mitosis is responsible for maintaining complex organisms and is therefore essential to organisms? Well, there are 2 main ways that I want you to know. I'm just going to list them. The first one is growth. Okay, when you are first conceived, you start out as one cell called a zygote. And you have grown into the complex organism you are today because of mitosis. That cell copied, and then those 2 copied over and over and over and over until you have billions and billions of cells that make up your complex body now. Okay, so growth is one way that is that mitosis is essential to us surviving. Okay, another way is repair. As we talked about earlier, if you have some sort of damage done to your body will try to fix that with mitosis. The surrounding cells that are like the tissue that was damaged will reproduce through cell division, mitosis, until that wound or whatever damage is done is healed. Okay, so growth and repair are the 2 major ways. Number 5, copy and paste a diagram of the cell cycle, or it looks like a pie chart, and tell me the number of chromosomes at each step. So here's what we're going to do. I'm going to show you how to do this. I'm going to go to insert, and I'm going to hit image, and I'm just going to search for one. So look here, I'm going to type in cell cycle. All right, that's a pretty good one, I guess. I'm trying to find one that's relatively simple. Yeah, let's just do this one. Yeah, so we'll hit, my video is in the way here, so I'm just going to move my face. So I'm going to select that, hit insert, move my face back. And so here is our diagram. And so what we have, this is going it's a different direction than maybe what you saw in your Stemscopedia. But here we have G1, S, and G2, which remember are all in interphase. Those are all the 3 steps of interphase. And then we have M, which stands for mitosis, which is where cell division actually happens. So your normal cells, your somatic cells, which means your everyday body cells, all have right now 46 chromosomes, 23 pairs, so 46 chromosomes. So at G1, you have 46 chromosomes. So 90% of the time, remember, is spent in G1 phase, which is the first step of interphase. And so during that time, your cells are just kind of doing their thing. They're doing their normal function with 46 chromosomes. Now, once you hit S phase, this is where DNA replication happens, where that DNA is copied ahead of time for mitosis. So if you go from 46 chromosomes and you double those, if you copy them over, then that gives you 92 chromosomes. So it'd be 46 here, 92 here. And at G2, remember, that's just where your cell is growing a little bit more and preparing to split in mitosis, but you're still have 92 chromosomes here because they were doubled in the S phase. And all through mitosis, you have 92 chromosomes, so that each of the two new cells can get 46 and 46, which is what they need to do their normal function. Now at cytokinesis, when the cell splits, that is when you would have two new cells, each with 46 chromosomes, and then both of those enter G1 again until it's time for S, and then they double. So the only one on this specific diagram where you would have 46 would be G1, but understand that's where they spend 90% of their time. All the other phases would be 92. Okay, so 46 here, 92 for these. Number 6, describe each of the 4 phases of mitosis and include what is taking place in each one. So the first one is prophase. So in prophase, your DNA condenses into chromosomes. It's one thing that happens because when they're not condensed into chromosomes, they're in this long spaghetti-looking stuff called chromatin. But when they're condensed into chromosomes and tightly bundled up, they're packaged very conveniently to be moved around and separated and split. And so it makes sure that they're packaged up and can go to the correct places within the cell when they condense into chromosomes. The nucleus dissolves, okay, because we got to get that DNA divided into the two new cells. So we got to break apart that nucleus so that it can leave the nucleus. And the spindle fibers form. Those are those fibers, remember, that are going to help move the chromosomes around. The next step of PMAT, so we have prophase, now we have metaphase. Metaphase is very simple. It is where the chromosomes line up in the middle of the cell. So they line up straight down the middle so that they're very easily divided in two. Anaphase is when the chromosomes, let me rephrase this, the spindle fibers pull the chromosomes apart to each side of the cell, okay? And then finally you have telophase, and it is simply the opposite of prophase, okay? So here the spindle fibers are going away, the nucleus is reforming, and the DNA is starting to relax back into that spaghetti-looking chromatin. Okay, now those are technically the 4 phases of mitosis. And once you get to the end of telophase, the cells kind of look like this, where they're almost pinched apart. You have what's called the cleavage furrow forms, where it's like an indentation between the two new cells. And then finally, after mitosis, cytokinesis happens, which is where the cells pull apart. Cytokinesis literally means cell movement. So that is where the cells actually move apart. Technically not one of the 4 phases of mitosis though. So really you just need to know PMAT, prophase, metaphase, anaphase, telophase. Number 7, if organisms from 2 different species produce the same protein, such as albumin, hemoglobin, amylase, whatever, what else would you expect these two to have in common? Well, you got to think. So let's say I'm using one cookbook and you're using a different cookbook, and we both happen to make the same food, the same kind of food. What does that tell us about those 2 cookbooks? Each of those cookbooks each have the same recipe for the food that we just made. So if we have 2 different species that are making the same protein, that tells you that their cookbooks or their DNA has to have the same set of instructions to make that protein. So like, let's say we look at hemoglobin in a pig, and we see that it's the exact same as the hemoglobin that's in us, then we know that both we and the pig share the gene or share the instructions for how to make that protein. So both organisms would share the same gene or sequence of DNA that codes for that protein. It's that simple. We both share the instructions in our DNA to make that protein if we can both make that protein. All right, number 8, genetic diseases such as Huntington's disease result in the inability to produce a certain protein. So what would be the cause of this? So let's say let's say let's use that same cookbook analogy. Let's say I've got a cookbook and it's supposed to have a recipe for cheesecake in it, and I want to go home and I want to make a cheesecake. And I get home and half the page is torn out, and I can't make the correct kind of cheesecake because I don't have the correct recipe. Well, it's the same thing here. So this genetic disease, like Huntington's, prevents your body from making a certain kind of protein. And so what would be the cause of that? Well, it would come from the instructions for how to make that protein. So it would come from some sort of miscoding or misreading of a DNA sequence for that protein, also known as a gene. Okay, so just a miscoding or a misreading of that DNA would prevent us from making that correct protein. What evidence would support a scientist's claim that a certain gene is responsible for holding the instructions to produce a specific protein? This is actually a pretty tough question. I'm going to do my best to answer it briefly and clearly. So let's use this analogy. Let's say you work in a factory, and there are 4 different machines that are used to make cars. Okay? Got to do a good job of explaining this. Now, let's say this is totally pretend. Let's say you cannot tell what any of these machines are doing. All you know is that when all 4 machines are on, a finished car pops out of the end of the line. Okay? Now, let's say someone came in and asked you that a certain machine, this is an analogy representing the gene, is used to make the doors of the car. How would you figure out which of those 4 machines it is? Well, if you can't see the machines, you can't see what they're making, all you know is that all 4 make that car. I think the way you could figure it out is turn off the machines one at a time. So let's say you've got all 4 machines running, you get a nice functioning car, it's got the doors on there and everything. You turn off machine number 1, let it run. The car pops out, it still has doors on it. So that what that tells you is that machine 1 is not responsible for making the doors, right? So let's say you turn that one back on and you turn off machine 2. You let it do its thing, make a car, the car pops out, and there are no doors on the car. That tells you that machine 2 is likely responsible for making the doors of the car. So let's use that analogy to try and explain what's going on here with number 9. So they're claiming that a certain gene is responsible for holding the instructions to produce a specific protein. They're making the claim this machine has the instructions to make that component of the car, or this gene has the instructions to make this specific protein. So how would we figure that out? Well, you wouldn't want to turn that gene off in somebody intentionally because that would be unethical. Instead, you would want to find someone who does not have that gene or has a mutated version of that gene causing it to not function properly. So you want to find someone who has that gene that is either miscoded, so they've got a mutation. I really wouldn't find anyone who didn't have that gene, but you want to find someone who that gene is not working, okay, because of a mutation. And then see if they can produce that protein. So for example, let's say someone wanted to find out if gene number 3 is responsible for making the protein albumin. You don't have to know what that is. It's just the protein I picked, okay? Well, you look at someone who has that functioning gene, and they can make albumin, but that doesn't necessarily tell you that that gene is responsible for it because all their other genes are also working. Instead, what you want to do is find somebody who that single gene is not working and see if they can still make albumin, and that would tell you whether or not that gene is responsible for that. I hope that makes sense. I usually do a better job at explaining that when I have kids in here with me, so I hope that makes sense. All right, number 10. I'm going to speed up because this video is going to be long. Describe the relationship between genes, DNA, and proteins. Very simple. Genes are segments or sequences of DNA that code for or hold the instructions for specific proteins. Okay, so you've got a gene for hemoglobin, you've got a gene for albumin, a gene for all your proteins. They're just segments of your DNA. Individual recipes in the whole cookbook that are used to make specific proteins. Number 11, explain how it is possible to have cells with different functions if all cells in an organism have the same set of DNA. Okay, so let's say you work at a restaurant. We're all about the analogies for this video. So let's say you work at a restaurant, and I want everyone in the restaurant, all of the cooks, to use the same cookbook. So they're using the same menu, right? But let's say you've got one cook, he's in charge of breakfast, one cook, he's in charge of lunch, one cook, he's in charge of dinner, and then the fourth one, he's in charge of dessert. They're all using the same cookbook. They all have the same set of information, but I want them to do different things. So it's possible to have cells with different functions, even though they all have the same set of information, the same set of DNA, because only certain sections of that DNA, which we call genes, are being read to produce the proteins needed for those specific, I can't spell, for those specific functions of the cells. Okay, so just like if you're working at that restaurant and your job is to cook dessert, you have all the recipes there to make breakfast, lunch, and dinner, but your job is to make dessert. So you're only going to read the recipes to make dessert. If your cell is a skin cell, it has all of the information there to be all of your other different cells, but its job is to be a skin cell. So it's only going to read the genes needed to make the proteins to make it a skin cell. I hope that makes sense. Number 12, explain using a real-world example how mutation can change the overall function of a cell. A gene mutation may not change the function of your cell. It could cause a silent mutation, which you learned about in the elaborate activity, but it wants to know how it can change. So specifically, a substitution mutation, or just one in which one letter of your DNA is swapped out for another, can cause the disease sickle cell anemia, okay, by changing the structure and function of hemoglobin. That's a real world example that you've learned about a couple different times in a couple different lessons during this unit. So this specific mutation switches out one letter, and it takes your hemoglobin that is supposed to just kind of free float around inside of your red blood cell, give it a nice round flexible shape, and it causes the mutation, the substitution, just swapping one letter out of billions of base pairs, swaps one letter, causes your hemoglobin to be built incorrectly, and it clumps together and causes your red blood cells to be sickle shaped, and that causes sickle cell anemia because you can't get blood to certain parts of your body, and those cells will die a lot faster than they're supposed to. And so someone who has that disease has anemia, has a lot of different issues. Okay, that's a real world example though. So number 13 says, write a scientific explanation of how DNA holds the instructions for proteins, for the production of proteins. I am not going to write this out for you. I'm rather just going to kind of talk and get you going, because it would take me a good little while to actually type out a CER for this. So what you'd want to do is make a claim first to answer this prompt. It says something to the effect of, you can even restate it. You can say DNA holds the instructions for the production of proteins. That's a spot on claim. Now, the tricky part is you've got to then back up that claim. So your evidence is you're going to list different things, different pieces of information that back up your claim. So you say DNA holds the instructions for the production of proteins. You've got to now back that up with info. So I would say transcription is one piece of info, and if you wanted to expand on that, you could say transcription is where DNA is copied into mRNA. You don't want to explain anything yet. You just want to type information on your evidence. Then you can do translation. Translation is where mRNA is translated into proteins. You could also say ribosomes make proteins. You know, you can just type information here. And then in your reasoning, you're going to type a paragraph. Remember, it has to be a paragraph explaining how each piece of your evidence supports your claim. So if you said transcription in your evidence, you would say transcription is the process by which your DNA is copied into a strand of mRNA this needs to happen because the DNA cannot leave the cell, so the mRNA takes the instructions out of the cell into the ribosome so that it can make a protein. And then you just continue to explain protein synthesis from there. The ribosome reads the mRNA codons, brings in tRNA that has the amino acids attached to it, and uses those amino acid building blocks to construct the correct protein based on the instructions that the mRNA copy from the DNA. Look at that. I just wrote a pretty awesome CER, I think. CERs, don't get overwhelmed with those. They're not super difficult. You're just following a logical progression of thoughts, okay? You're answering one question. You're just explaining to me how you knew the answer to that question, okay? So don't get too overwhelmed with that. There are great CERs that are super long, and there are great CERs that are super short too, okay? It doesn't have to be long to be a good CER. So I hope that made sense. I hope that clarified some things for you guys. I feel like I moved a little fast, but you can always go back and rewatch the video, and you can always go back and study your Bellworks and those Stemscopedias, okay? All right, if you need any more help, let me know. Email me, call me. Thanks, guys.